Namaskar दोस्तों, welcome to channel Toxicology Unknown. Today in this video, in continuation with the ICH Aid guideline, we'll be discussing the last video we discussed few questions. So here we'll be discussing each and every section in the guideline. So subsequent 2 to 3 videos I will make only on the understanding the every section in the guideline. Okay, so no questions will be discussed in this video. Okay, so the first what we can understand from this particular section is what is the objective of the SAID guideline. So first question also in the previous video was the same thing, objective. So that particular section 1.1 we'll see. So objective of this SAID guideline, immunotoxicity studies for human pharmaceuticals. The objective of this guideline is to provide recommendations on non-clinical testing, approaches to identify compounds which have potential to be immunotoxic. So, if you are developing a certain new molecule, and if that new drug is going to trigger the immune system, whether it is triggering the leukocyte count or T helper cell, like that. So, those, if it's having certain potential of concern to affect the immune system, so that type of molecules, that type of developing drugs, you have to consider ICH guideline to understand whether additional toxicology studies are needed or not. Okay. Second one is this particular guideline, ICH guideline, it is giving you guidance on weight of evidence decision making approach for immunotoxicity studies. So what is this weight of evidence also we'll discuss in the next slide. So it should be unintended. The main purpose of this immunotoxicity is for the purpose of this guideline defined as unintended immunosuppression as well as enhancement. See, this is unintended immunotoxicity as well as immunoenhancement. Drug-induced hypersensitivity as well as autoimmunity, they are excluded from this ICH guideline. We have separate guidelines for the understanding. So drugs which are triggering the autoimmune system or which are causing hypersensitivity reaction like beta-lactam antibiotic penicillins, we have separate guidelines for those type of category of chemicals. Okay, objective is to identify potential immunotoxic effects of pharmaceutical candidate early in the drug development to support the human risk assessment. Okay, and second one is principles is immunotoxicity evaluation should be part of the overall safety assessment. Whatever the toxicology battery of toxicology studies are there, no, it should be a part of overall part of this safety assessment. Second one is weight of evidence approach is recommended as per ICH-8 guideline. Third is testing strategy should be based on mechanism of action. What is the developing drug, whatever the candidate which you are developing, what is the mechanism of action, structure, duration of the treatment, whether it is 6 months treatment, 1 month treatment, or 3 month treatment, and clinical relevance. Use of validated assays and appropriate controls is essential. Interpretation should consider findings on the context of the toxicity data. Okay, that was about the section 1. Now we'll discuss what is the background of this ICH-8 guideline. So this is the section which we have taken from the guideline. That is 1.2. We are discussing section 1.2 background. So from this particular section, what we are going to understand is what is the background of ICH-8 guideline. Evaluation of this potential adverse effects of human pharmaceuticals on the human immune system should be incorporated into the standard drug development. So whatever the battery of toxicology studies are there, 28-day, 3 month or gene tox study or reproductive study, in that immunotoxicity study should also be a part of the um, incorporated in the standard drug development. You have drug development, different phases will be there early in the drug discovery only. Toxicity to the immune system encompasses a variety of adverse effects. So, what are those adverse effects we will see? These include suppression, that is immunosuppression, and immunoenhancement of the immune response. So, in the immunotoxicity, they're going to read immunosuppression and immunoenhancement. What is this immunosuppression? It leads to decreased host resistance to infection agents or tumor cells. For example, when you are doing organ transplant also, okay, intentionally you are taking immunosuppressive drug so that it will not reject your transplanted organ. Okay, so whatever the new drug which you are developing, that that should not that should not cause immunosuppression. It should not be. It should be unintended effect. If it is unintended, it is going to cause immunosuppression, then that will be considered an adverse effect. Okay, lead to decrease in the host resistant to infection agents or tumor cells. Thereby, you will get tumors. You will get infections also. You will be getting because of these drug which you are taking this new developing drug. Second one is immunoenhancement can exaggerate autoimmune disease or hypersensitivity. Okay, so there are certain drugs if they're going to trigger and it is going to cause autoimmunity or hypersensitivity drugs. So these one. So we'll see what. So what are these? We'll see suppression of the immune response can lead to decreased host resistant to infectious agents, that is virus, bacteria like that, or tumor cells. Enhancing the immune response can exaggerate autoimmune disease or hypersensitivity. Drug or drug protein adductor. This is your drug as well as drug protein adductor might also be recognized as a foreign. Your body will identify this drug protein adduct as a foreign, as a xenobiotic. Okay, can recognize as a foreign and stimulate an anti-drug response. Immediately, as a defense mechanism, your body will start showing anti-drug response. Subsequent exposure to the drug can lead to repeated exposure. Second exposure, it might lead to hypersensitivity allergic reactions. Much of the science and method development and validation efforts in the past have been focused on evaluating drug development candidates for their potential for their either immunosuppression or contact sensitization. Previously, before the guideline came into existence, the focus during the drug development was only to address these things, whether the candidate is causing immunosuppression, whether the developing drug is causing contact sensitization. No standard approaches for human pharmaceuticals are currently available for testing for the respiratory, whether the newly developed candidate whichever you're testing, no, whether it is going to trigger the respiratory sensitization, whether it is going to cause systemic allergenicity. These points have not been addressed previously. Okay, drug-specific autoimmunity. Testing for these endpoints is not currently required in any region. Any region means either it may be LATAM region or US, Europe, Japan. Okay, there are no regional differences in testing approach to skin sensitization. So pause the video here. This paragraph is little bit tricky to understand because it is you have to understand immunosuppression, immunoenhancement, immunogenicity, immunotoxicity. So these terms you have to understand. And another thing what you have to understand here is regarding skin sensitization. So what are the, pause the video and go back and check what are the different essays, what are the tests in the given in the even in the case of OECD guideline. Remember the numbers, whether what is the number 480, 470, whatever the numbers are there, no, which are responsible. For example, OECD 406 is a GPMT essay. So like that LLN essay, you see what are the different types of skin sensitization essays are there that you see. Okay. Now we'll go to, in addition to 1.2 background only. So the continuation of the paragraph. So what is the, in addition to what is the additional, what is the background of this ICH guideline, we'll see immunosuppression, immunoenhancement. These two terms you have to keep it in mind when you are reading ICH guideline. Can be associated with two distinct groups. What are those? We'll see. Drugs which are intended to modulate immune function for therapeutic purposes. Okay. Intentionally, these whatever the developing molecule, it is going to have it is going to modulate, it is going to work on your immune function, immune organs, whatever. Example, to prevent organ transplant rejection. As I told you, if you're there are immunosuppressive drugs are there. If a patient undergoes liver transplant or kidney transplant, their physician will intentionally prescribe immunosuppressant so that the newly placed organ, transplant organ, will not reject your body. Okay, where adverse immunosuppression can be considered exaggerated pharmacodynamics. Okay, it should have it, whatever the drug which you are giving in such cases in the organ transplant patient, it has to suppress the immune system, but it should not show exaggerated pharmacodynamic. Excessive immunosuppression will also will cause adverse effects to the patient. That you have to keep it in mind. Little bit confusing this statement, but try to understand. Second one is drugs not intended to affect the immune function. You are developing a certain drug. For example, you are developing anti-hypertensive or anti-lipidemic drug. It should not trigger the immune system. The target organ is not immune system. If it is going and affecting, then there is a cause. What is we'll see. Drugs not intended to affect the immune function but cause immunotoxicity due, for instance, example, to necrosis or apoptosis of the immune cells. What are those immune cells? T helper cells, leukocytes, whatever they're there, or interaction with the cellular receptors shared by both targets. So whatever the helper cell or T, whatever the immune cells are there, and whatever your investigational drug, the drug which you are developing, both are having same targets shared by both target tissues and non-target immune system cells. Okay, bit confusing. Stop the video and again you read this point number one and point number two. Let's read below paragraph. Anti-proliferating agents used to treat cancers. Anti-proliferation means in the case of cancer, what is happening? The cells are rapidly multiplying. So what you are trying to give, you are going to give a drug which is going to stop the cell division. That's anti-proliferative agents used to treat cancer are an example of drugs that produce unintended immunosuppressions. In such cases, what do you want? You want to cause immunosuppression, but not exaggerated immunosuppression. This word you have to remember, exaggerated. You keep coming across this word, exaggerate. Whenever you are writing report writing also, no, you'll come across this word exaggerated pharmacodynamic response, exaggerated pharmacological effect that you'll see. Okay. In such instances, adverse findings in non-clinical studies are a predictive of human immunotoxicity in a rather straightforward manner. So, in such cases, who are the scientists who are developing the certain drug? No, he already knew this is going to definitely cause immunosuppression, and this is exaggerated immunoresponse. Okay, in such cases, adverse findings in non-clinical studies are predictive of human immunotoxicity. Already, the researcher knows this is the explanation for this type of why this particular drug is behaving or why this particular test species is showing these effects. Next one we'll see that is specific assays to determine immunotoxicity are probably not available in drug risk assessment since the target tissues are usually rapidly dividing cell types. So very difficult to identify biomarkers in such cases, such as bone marrow derived immune system progenitor cells. So here you can pause and go back and check in the bone marrow. There will be different erythropoid, the lineage will be there, no different stages of the cell, how the RBCs form, that you have to remember, progenitor cells. Hence, the adverse effect on the immune function can be predicted based on the pharmacological activity can usually be reliably evaluated in non-clinical studies. In such cases, since you do not have a proper biomarkers, here the cells are rapidly dividing. So in such a case, how you can depend? You can depend on the pharmacological studies. The results from the pharmacological activity can usually be reliably evaluated in this non-clinical studies. Next, we'll see for other types of compounds not intended to suppress immune response. There are certain drugs, for example, as I told you, antihypertensive you're developing or anti-diabetic drug. Distinction between exaggerated pharmacodynamics and non-targeted effect can be less obvious. As an example, here they have given in the guideline, ICH guideline, you're developing some anti-inflammatory compounds have effect on the certain innate immune functions but do not necessarily affect the adaptive immune response. So you have to stop the video and see different types of immune systems are there. Innate immunity, what is this one? Adaptive immunity, humoral immunity. So please read, then only you can understand what this particular point is. Cell-mediated immunity, that is T cell proliferation, innate immunity, phagocytic activity, humoral immunity, that is antibody production, right? So we'll see another section 1.3 we'll discuss here. What is the scope of this guideline? What this ICH ESSAID guideline is going to cover. Okay, what is the scope of ICH ESSAID guideline? So we are reading section number 1.3. Okay, the guideline is focused on providing recommendations on non-clinical testing for immunotoxicity induced by human pharmaceuticals. If you're developing a new molecule and the drug is causing immunotoxicity, so what kind of toxicology studies you have to do? This is the only thing that you have to remember. Then only you have to refer ICH ESSAID guideline. Otherwise, if it is not causing, if there is no concern, you have to stick to ICH S4 guideline. If it is a biotechnology derived product, stick to ICH S6 guideline. If the drug is in the later stage, treatment of the patients at the last, last survival is there, end stage survival, I said S9 guideline. Like that, you have different guidelines for different. If it is a oligonucleotide, you have to go for ICH S12 guideline, like that. Okay. So it is restricted to unintended immunosuppression. You keep coming across this word, unintended immunosuppression, unintended immunoenhancement, excluding allergenicity and drug-specific autoimmunity. So these things are not covered under, these are out of scope of this particular guideline. Next, we'll see the guideline applies to new pharmaceuticals intended for use in humans as well as to the marketed pharmaceuticals. So always when you're reading ICIT S5 guideline, ICIT S9 or S6 guideline also, there is small variation in the type of toxicology studies needed for the phase 1, 2, 3 trials as well as for different from that of marketing trials. So these small variation and certain times with respect to region also. What kind of studies need to be submitted for Japan or for the US or Europe also, a little bit of difference will be there. Okay, so the guideline applies to new pharmaceuticals intended for use in humans as well as to the marketed pharmaceuticals. So if you're planning to market your drug, so what kind of studies are needed? Okay, proposed for different indications. So for example, you have developing a certain drug X for this type of indication. For example, you're developing it for anti-hypertension. Now we want to include some diabetes also for that indication, or some other weight lowering also if you're trying to add another indication to the same molecule. So for different indications or other variations on the current product label, or there is a difference in the strength or different in the dosage form. First you develop capsule, now you develop onto tablet, or certain times you develop into solution, syrup, like that, in which the change could result in unaddressed because of the change in indication, because of the change in the formulation or dosage strength or like that. If the because of these changes, slight changes also, if it is going to trigger your immune system, then that also you have to take into consideration, which change could result in unaddressed and relevant immunotoxicity issues. In addition, the guideline, so in such cases also you have to go for immunotoxicity studies. That is what it is saying. In addition, the guideline might also apply to drugs for which clinical signs of immunotoxicity are observed during clinical trials. Initially, during preclinical trials, you didn't find any kind of triggering or any red flags you didn't find. But during clinical trials, when you are starting phase 1 or phase 2, if you are found and following approval to the market, at that time also additionally immunotoxicity studies you have to include. Okay, the guideline does not apply to biotechnology derived products because for biotechnology, for biologics, we have separate ICH S6 guideline, preclinical safety evaluation for biotechnology-derived pharmaceuticals, S6R1 revision guideline. Okay, and the other biologicals. So we have, as I told, no, for oligonucleotides, we have ICH S12. For for uh, for um, for vaccines, your separate guideline will be there. So like that, separate guidelines will be there. Next, we'll see existing guidance, the document on sensitization or hypersensitive remain in force and are not affected by this document. So whatever the information with respect to sensitization, hypersensitivity, we have separate guideline, which is not affected by this particular guideline. It is beyond the scope of this guideline to provide specific guidance on how each immunity, as I told you, know, you have to read what is adaptive immunity, what is humoral immunity, what is cell-mediated immunity. If so hypersensitive, you have to remember always type all the four types and types of immunity also. General methodology guidance is given in the appendix. So in the subsequent videos, we'll discuss what is appendix, that we will see. In appendix, what are the different essays what they have given. And we'll also see there is a decision tree diagram. This is the decision tree diagram also we'll see. The next question is, what is the overview of ICH essay guidance? So what is it is covering? In section 1.4, we will see the general principles that apply to this guideline are all new human pharmaceuticals should be evaluated for the potential to produce immunotoxicity. Okay, whatever the new molecule, if I am a researcher, if I am a pharmacologist, if I am a toxicologist, if I am developing a new molecule, so what are the sorry, what are the potential to produce immunotoxicity? If my chemical has some red flags, whether it is going to cause immunotoxicity, then you have to include this type of endpoints also in the drug development. This is the one thing. Second one is methods include. What are the methods? Include standard toxicity studies, STS, that I told you, battery of toxicology studies. Additional immunotoxicity studies conducted as appropriate. Based on decision tree, you have to decide whether we have to need additional study or not. Whether additional immunotoxicity studies are appropriate should be determined based on weight of evidence review of factor in section 2.1. So 2.1, previous video also we have discussed. The next video also will discuss in detail. So if any points are missed, the description of the guideline below will follow the recommended decision process in immunotoxicity evaluation as shown in flow diagrams. So whenever certain diagrams are there, you have certain guidelines are there, safety or quality guidelines, you have to read the tables, decision tree table, you have to read, and there will be some summarized tables will be there. Those also you have to always, examiner will ask question from those tables only. More detailed description of the testing methods are discussed in appendix. What is this appendix? We'll see in the next video. And very quickly we'll go through the decision tree. This is figure 1. So flow diagram offer recommended immunotoxicity evaluation. So all human pharmaceuticals, so all small molecules, conventional traditional molecules, non-biologics. So biologics we have separate ICH-S6 guideline, they're excluded. If I am a researcher, if I am developing a new drug and that new drug is small molecule, then this is what you have to do, decision matrix. Second, in the decision tree is section 2.1. Identify the factors to consider. We have to see, is there a decrease in thymus weight, decrease in the whatever the immune system organs are there, no, thyroid weight, or there is a decrease in the leukocyte or granulocyte count, those or T helper cell count, like that. Weight of evidence, different things you have to see. There is checklist will be there. Okay. Weight of evidence review warrants additional immunotoxicity studies. Based on this weight of evidence, you will decide yes or no. You will decide. If it is no, additional non-clinical immunotoxicity is not needed. Stop. There is no more study is needed. Next, if there is a potential concern, if from the repeat dose toxicology studies you find that there is a decrease in the leukocyte count or increase or decrease in the or increase in the organ thymus weight or thyroid weight, like that, then what you have to do? You have to conduct additional immunotoxicity studies. What are those studies? TDRSA, immunophenotyping. That we'll discuss in next video. Okay. Significant changes observed? Yes. Here after that, if it is there is no further non-clinical immunotoxicity not needed. Yes. Sufficient data for risk assessment, risk management. No. Consider further immunotoxicity study. So like this, you have to take your molecule, see in the decision tree where do I stand. In the next videos, no, we'll take one example also, one particular drug which has been approved and for which is there in the market and for which immunotoxicity studies have been conducted. So how the study design is there, how many animals you have to take, what are the different parameters we'll be checking in the case of immunotoxicity, because many of us will know what is the 28-day tox study design or 90-day tox study design. So we'll discuss with respect to one immunotoxicity study. Okay, with some example also we'll discuss. Okay, thank you so much. Be curious. For any questions, you can always write a mail to arya.priyanka18@gmail.com. All the best for your exam.